Thanks for reminding me. All right, so proteins are amino acids composed of amino acids. They're the monomers. So there's a structure of one. I just draw this like this. That's a carboxylic acid. At one position, there's a hydrogen at this position. This R is a functional group. It changes for the 20 different amino acids. And there's a nitrogen group over here. And it's really the nitrogen group that makes proteins protein, as well as a functional group. But they call it a functional group because depending on how these are linked together, it'll determine how the molecule folds and takes shape. The shape of the protein is how it'll be functional to your body. Okay. So what you can do is you can take these amino acids, you can link them together. And there's a bunch of different kinds. Maybe there's 20 different kinds. They show you a few here. You're not responsible to know all the 20 different amino acids. They show you a handful here, but you should at least be aware that there are 20. And you should be aware that when you link them together, as shown here, two amino acids linked together. They're linked by a covalent bond called a peptide bond. Okay. Jeez, I keep kicking everything there. Give me so much space. Okay, peptide bonds. Link amino acids together. Oops. I think what's important for you in being aware that there are 20 different amino acids, some amino acids are hydrophilic and some are hydrophobic. Okay, I'll kind of get into the hydrophobic thing. That's really important. Generally what we say in biology, if you're a molecule that kind of has more hydrogen and carbon, you're more water-hating, kind of like non-polar. Molecules with more carbon, hydrogen, tend to be non-polar. Or hydrophobic. But molecules that have other organic elements like oxygen or nitrogen, if you look at this here, like this orange group, if it has ring structures and a lot of carbon and hydrogens, this as a group is kind of more hydrophobic than say some other groups like, I don't know, like say this one, we have nitrogens at the end, or this one. Okay, that's the general rule. So I give this as a general rule and move on. And when proteins kind of fold and are formed, the protein is transcribed and it's translated by the cell. It can make a diversity of different proteins. And I just gave you a laundry list of all the ones that come up for A&P students a lot. And I want to define them today. Okay, so I'm going to spend some time defining biological proteins, since they come up in chapter 3 a lot. Now, eating protein is a lot more fun than learning about the proteins that cells make. That's why you eat the proteins, right? There's all the stuff your body needs. Whether it be from plant sources or animal sources. Animal sources are the more cancerous. Plant proteins. Plant proteins are better proteins. That's kind of a misconception that's out there. People think, I gotta eat meat, protein. Not just recycled protein. Do you know that plants are the ones that actually originally make the OG protein? They can extract the nitrogen from the soil. Animals can't do it. So plant proteins are way better. Way, way better for your health.
Excuse me.
No, it's okay. Okay, so enzymes are proteins. Basically, in chemistry, you might learn these lower the activation energy. Sometimes abbreviated E subscript A in books. Oh, so basically what that means is it'll speed up chemical reactions. It's more likely to happen. So in biology, one that's important in physiology that I teach a lot, there's an enzyme called, just to give you one example, Carbonic Anhydrase. What that does is we say this enzyme, it hydrates water, hydrates CO2, as you like to say. So CO2 plus H2O. These two molecules combine, and it would be very slow without an enzyme, because these are two pretty inert molecules. They don't really want to interact with each other. But what they would form is that carbonic acid. This is a weak acid, which means it can partially dissociate in a solvent into HCO3 negative charge. Gives off a hydrogen ion. So this is bicarbonate. Bicarbonate is hugely important when you study renal. And this reaction can go both ways. And right here is where the carbonic anhydrase will speed up the chemical reaction. Okay, that's really important in human physiology. Just give you one example of an enzyme. Now, cytoskeleton is the next one. Cyto means cell. And I know whenever you say skeleton, you think of the human bony skeleton. But in this one, it provides cell structure, some rigidity. To intracellular organelles. Well, one we'll study in chapter 3, again, just to give you an example. Tonal fibrils of desmosomes. I'll talk about desmosomes later. They basically help link cells together, and tonal fibrils are the protein that could be considered a cytoskeleton. So cytoskeleton, they're basically protein fibers inside the cell providing rigidity. Okay, that's literally what it is. Fibers. Protein fibers inside cells. All right, hormones like insulin. Hormones can be proteins. They can be steroids too. But a lot of hormones that you'll learn in endocrine are proteins. Now hormones, when they're synthesized, we call them chemical messengers, and they're secreted in the bloodstream. That's kind of what I mentioned about insulin last time. So, synthesized by cells of endocrine glands. And secreted in the bloodstream. Secreted into the bloodstream. And I already gave you examples in previous lecture, like insulin, right? That's an example of one. Okay, antibodies are proteins. You fought a cold or flu this cold season, your antibody titer is up. Antibodies are, the protein name of it is called immunoglobulins. I mean, usually we illustrate them on the grease board as this Y shape, because they have these specific and non-specific regions. Well, you don't have to know that now, but basically understand these will actually fight foreign invaders for you. They're also secreted in the bloodstream. They're just proteins. They're secreted by the B limbs. I guess you should know that. These are antibodies, or immunoglobulins. Secreted by the B limbs. So imagine you have a B lymphocyte. Cell. And actually, it has as its membrane receptor an antibody. It's membrane bound. And then you catch the flu from your spouse or someone in class or something. You get past all your defense mechanisms to get into your body. The little red dots illustrate pathogen, bug. And it happens to be this clone. You got millions, you got billions of these clones of your white blood cells. It's actually specific for this particular bug, but no other bug. Okay, like this one. So you get a different bug from your kid. The blue one. This one can't fight it. It's not specific for it. This is specific immunity here. But maybe you have another clone that does. When this one gets activated by the red one, it can mount a defense. You'll actually proliferate this clone. And it'll become a big cell. It'll increase its cytoplasm. It'll become this immunoglobulin antibody making machine. And it'll secrete these into the bloodstream to fight your cold. Maybe take a couple weeks to get better, maybe sooner. And they secrete in your blood. And what these do is, these will bind up all the red dots. And what we say is antibodies aren't the murderers. They're accessories to murder. What they do is they'll kind of sop them up, kind of hold them. And then other cells, like macrophages, will come along and phagocytize them. Okay, but they're very important for immunity. Basically 50% of your immunity, in my opinion. The other 50% are the T cells. But anyways, they're proteins. Receptors are proteins. Super important. It's how cells can respond to things. Cells can't do anything if they don't have a receptor to bind. They have to bind something. Sometimes textbooks will use the term ligand. It's just something binds the receptor. Like this bug bound the antibody receptor. So in this example, the antibody is the receptor, and the little red bug is the ligand. This is kind of generic terms. But they can be proteins. They're usually membrane bound, or they could be intracellular. So if you have a cell, here's the nuke. It depends what you want to bind. If the thing you want to bind, say this structure here, this blue square, it wants to target this cell. It's supposed to. But the thing is, it's a protein, and proteins don't diffuse well past the cell membrane. So this cell, when it makes the receptor, it has to present it on the cell surface. Okay, so that's an example of a cell receptor. So it could bind something that's not water soluble. You know, but what if it is? What if, like, I don't know, this is a lipid. Lipids really have a tendency just to get through, because membranes are made of lipid, and like dissolves like, so this can kind of go right through. So maybe that can bind its receptor in the cytoplasm. So receptors can be cytoplasmic, right? So that little shoehorn is, that little U shape is the receptor for that. They could be in here. They don't have to be. They could be in the nucleus. Maybe it's supposed to turn genes on. So maybe it could bind, go right through that membrane, and bind its receptor in there. Okay, so you know, receptors can be membrane bound, or they can be nuclear receptors. They can be intracellular receptors. All right, skeletal muscle. We have a whole chapter on that. We'll learn about the structure of skeletal muscle. Muscle's protein. People generally know muscle's protein. Now, at the molecular level, we'll learn about actin and myosin. Those are the two main muscle fiber proteins that we'll learn about in anatomy and physiology. So I'd write those words down. Actin, myosin. These are thick filaments. These are thin filaments. Okay, you'll learn that later. Actin are the thin ones. Thick. That's kind of what your muscle's made of. Well, we'll get into what muscle is. It's chapter 9. All right. What we do more for chapter 3 in many chapters of this book is when you study cell physiology. It's good for students to be familiar with the terms channels, transporters, pumps. I noticed that those terms are used a lot. Well, it's kind of understood that you know what they mean, so I should take the time to explain it. So like I said, proteins don't get through the cell membrane very well, so they need help. They need another protein structure to be embedded in the cell membrane to allow to facilitate its diffusion through the cell membrane. In fact, they call it facilitated diffusion. So let's say you got a channel with a pore. This is protein, this blue thing. There's a little pore right down the middle, and this is embedded in a lipid bilayer cell membrane. Let's just call this out, let's call this in. Now, if you're a molecule that's specific for this, call it a channel. Channels usually have a pore. That's kind of my definition. They're kind of always open. They have a pore. Right there. Maybe it's for water, maybe it's for something else. The point is, it can diffuse through that if it's in high enough concentration. So let's say you got more out than you do inside. I mean, the rule of diffusion is you just kind of go down your concentration gradient. So you'd flow in. In this example, I've set it up. The blue would flow in until you get some kind of equilibration. Okay. So that's a poor, I guess you would call this facilitated diffusion, if we want to name the process. So I have a question for you. If the channel's not there, does the blue molecule diffuse? Yes or no? If the channel is not there, can the blue molecule diffuse into the cell? Yes? That's a yes-no question. Think about it. The answer is no. This molecule is probably, it can't diffuse past the, maybe it's polar. It's non-polar could get through, polar cannot. It can't get through the cell membrane by itself. It needs a protein to facilitate its diffusion. Okay? That's the whole point of it being called. And what does it mean to be facilitated? It means you need help, right? Like some of you who are waitlisted and I added you, I facilitated you into this class. You can't just be here because you want to be, like the people I turned away. Right? This is facilitating the diffusion of the blue molecule. Okay, well anyways, channel, okay, what's a transporter? Okay, same kind of thing. Any questions on facilitated diffusion? Because that's important, that's coming up again. All right, so I'm trying to draw something that will illustrate my point of what a transporter is. Well, my understanding of what a channel is, it's just got a pore in it. There's like a hole punched through it. That hole can let things in, but only if it's the exact fit for that molecule. So it's very selective, okay, what it lets through. Transporters, sometimes they bind their ligands and they change shape to allow diffusion in. So maybe it's like this red ball fits that shape, that's the ligand. Okay? But maybe this transporter transports other things too, along, so we say like along for the ride. Like maybe another molecule. And when they both bind, maybe this will change its shape to allow them in. I don't want to like keep redrawing, it takes too much time, but the idea is you would co-transport both of these molecules into the cell. Okay, so maybe they both get in. The red molecule and the blue one. And it could be a thing where they're both still following their concentration gradients. Let's say, just for simplicity's sake, just to be consistent, they are in this example. So there's more out here, less in here. And same thing for the other one that's being co-transported with it. So these are all proteins. The red balls, the blue triangles, and the molecule that's transporting them. Again, it's the same question. Can they be transported without the transporter, this red thing? The answer is no. You need the transporter. So this red thing I drew is a transporter. Whenever the word pump is used for a cell organelle, it means energy is being expended, usually. I haven't seen an example where the word pump was used and ATP was not used. So usually they're illustrated just like a simple circle in the textbook. What pumps do is they'll pump a molecule against its gradient. So I'll just say pump against a molecule's gradient. So that means you'll need energy energy expenditure. So here you got more on the inside, but this is like where you actually want to pump against it, go from low to high, usually high to low. And so to accomplish that, the cell needs to, usually it's ATP. Usually how it's illustrated in books is it'll kind of go like that. And this molecule can hydrolyze the ATP into ADP, an inorganic phosphate. Oops. Okay, that's how a book would illustrate it. So what's really happening is, it's like ATP is like the book's always called the cell's currency. It's like how much money you got in your wallet. Currency, literally. If you have no money in your wallet, you can't buy anything. For a cell, if it has no ATP, it's probably dead. The tissue's probably dying. Or, maybe you need to rest and eat or something. But anyways, high energy phosphates, what it is, it's like we illustrate it as 3 Ps. Now that last bond between the last P carries the most energy, and I can't remember the number, how many kilojoules it is or whatever. But the point is, if you're a molecule and you have an enzyme, say for example ATPase, which would be an enzyme that hydrolyzes ATP, and you break that last bond, energy is released. Okay, and that's the energy that could be used to transport that molecule against its gradient. That's basically what you need to know about pumps. So before I move on, any questions on those 3 things? The channels, the transporters, and the pumps. We'll have a specific example of this when we get to the next chapter. Like for example, you got heartburn and your doctor gives you a Prilosec, and your doctor will tell you it's a proton pump inhibitor. This one, what's a proton? Hydrogen. You're trying to inhibit it so you make less stomach acid.
So,
yeah.
What happens to ADP? Does it just float around until it finds another phosphate, or does it?
No, it can be regenerated into ATP. Yeah. Float around, I guess if you want to think of that's okay. Yeah, it's in the cell, and you have to regenerate your high energy phosphates to make more ATP later. Let's see. Oh, one more. Hemoglobin and Myoglobin. Any more questions? ADP does come up a lot. I mean, it does more than just wait to become ATP again. It has some function. It could be a cell signaler. It does have other functions besides just being the little brother of ATP. So you got ATP and you have creatine phosphate. High energy phosphates are really important for cell activity. The only way the cell can do things is to break bonds of high energy phosphates so energy can be released. I mean, that's what your food is. I mean, when you eat food, why do you eat it? Because you're breaking down the food stuff and you can actually generate ATP from it. Okay, and we'll actually talk about that today. Hemoglobin and myoglobin are proteins that bind oxygen. simply put. So it can be transported in the blood. Now, in myoglobin, myo means muscle. That's the protein in muscle that can bind oxygen. Hemoglobin, hemo, that refers to blood. So that's the protein in your red blood cells that can bind oxygen. That's basically what you need to know. So that's enough time on the different kinds of proteins. Really what this chapter tries to get students to understand is the level of structure. The level of structure means, let me just define it. There's primary, secondary, tertiary, and quaternary. And, well, here's a picture of the primary structure of protein. Protein structure. That's simply the sequence of amino acids in the polypeptide chain. I'll just kind of write it. That's the best way to describe it, the definition. The sequence of amino acids in the polypeptide chain. And they don't even give specific examples. They just say amino acid, but there's 20 different ones, right? And they're just in some order. Now that order is really important because the order, the sequence will determine the folding. Okay? And that determines the structure and the function of the whole thing. The sequence determines the folding. Those functional groups, it's just atoms interacting with each other. And it's like they fold in different shapes. So the first step is to know, well, what's the order? You heard of like a mutation? A mutation is this sequence is out of whack. Okay, maybe there's one missing. Maybe it's a single amino acid that's missing. Or you just substitute a wrong one. Okay, some mistake. And that protein probably will be non-functional. So the sequence is important. Now, they fold in basic shapes. So we usually give alpha helices and beta pleated sheets as examples of how they fold. That would be the secondary structure. Sequence determines folding into probably one of these structures, alpha helix, beta sheet. And you can see the illustrations there. I won't try to redraw them. That's what they're called. Now, notice the structure is kind of maintained by hydrogen bonds. That's one thing I'll point. It says it there. Structures maintained by hydrogen bonds. If you forget your type of bondings from chemistry, I'll review some of the basic ones towards the end here, but for now, just kind of note that. Now, so what you've got here is the sequence, one chain, one polypeptide chain. Each of these sheets or helices, you're still like in the one chain thing. Now, when you get to what they define as tertiary, still one chain, but what it is, it's like maybe a bunch of alpha helices and or beta pleated sheets together in series, but it's still one singular chain, just super long. It's just folded and forming some structure. So the definition is, it's still one polypeptide chain. maybe many alpha helices and or beta pleated sheets in a row. So maybe this is alpha helix, and then maybe right next to it is a beta pleated sheet. And maybe this is another alpha helix, followed by another beta pleated sheet. But that's all one polypeptide chain. It has a start, and it has an end. And to maintain this shape, this random shape that I've drawn, maybe there are other kinds of bonding that will help it maintain this tertiary structure. Maybe there's hydrogen bonding here. H bonds. There are stronger bonds called disulfide bonds, SS. That kind of takes more energy to break those bonds. Disulfide. Know those. It's tertiary structure, but it's just one chain. Now, many proteins we'll study have, you take more chains, more than one chain, you put them together, that will be a quaternary structure. Quaternary. Quaternary. Pronounce that right. Where, by definition, more than one polypeptide chain. Okay. Maybe you got another one of these that's similar. They give an example of prealbumin. Now draw another one. Helix. Helix, I don't know, I don't want to keep drawing forever. But just as an example, maybe that blue polypeptide chain is bound to the black one with the disulfide bond. Okay, something like that. Disulfide bonds are very hard to break. If you work with proteins in the lab, like what I did, one of my first jobs as a grad student, I had to run gels with proteins, and I had to break those disulfide bonds. I used a chemical called beta or capital ethanol. It was the worst thing I ever smelled in my life, and I've smelled like dead bodies. Okay, this is right there with dead bodies. Well, anyways, the point is, you need strong chemicals break these bonds. They're very strong. So let's think for a second. Look at the pictures based on what I explained. Look at the multiple choice question. You got a few things to choose from, what I just defined. What do you think A is? What do you think A is showing you? Probably secondary. It is secondary because what structure is A? It is secondary, but what is A? Is a helix, which I defined as secondary. So then what's B? Tertiary, because it's one polypeptide chain. They tried to show that to you by making it one color. And it's a bunch of helices in series in a row. So then what's C? Quaternary. They use color to show different polypeptide chains linked together. This is probably hemoglobin. Okay, well, anyways, that's the structure of proteins. Now, to review bonding, since it does come up a lot, I'm already mentioning it. This should be review. It is review. You all took chemistry. I got to update my prereq list, but suffice it to say, you took chemistry and you learned this. Okay. This is an ionic bonding review. An ionic bond involves the transfer of electrons the resulting ions have an attraction. That's the bond. And they give the good old sodium chloride as an example. You know, the outermost electron ring, it's being transferred from sodium to chloride, giving sodium a full positive charge, giving chloride a full negative charge, and hence the attraction. The resulting ions are attracted to each other. Okay, so that would be sodium chloride together. And, well, I mean, I should mention covalent bonds. Covalent bonds are when two atoms are sharing electrons. They share an electron pair. I'm not going to get into details of it, but you should know the difference between if you have equal sharing or unequal sharing of electrons. Because if it's equal, you got a pretty inert molecule that doesn't do much biologically. Equal sharing usually means you got a nonpolar molecule. I'll give some examples there. I mean, O2 is like pretty inert, doesn't do anything. There's no net charge around that molecule. Unequal sharing, you know, creates this dipole moment. Here's, always give water as an example. Unequal sharing, you create what I have there, this kind of polar covalent bond. And they give water as an example, the H2O. Unequal sharing means the electrons are spinning around one more than the other. So for example, it has to do with what's in the contents of the nucleus of the atom. So this one kind of pulls the electron, the oxygen pulls the electrons closer to it. They spend a little more time spinning around the oxygen, giving not a full charge, just a partial negative charge, not a full negative charge. And that's where they put the little delta sign. And that's kind of the dipole moment. So that means the other hydrogens have a little positive dipole moment. So water is the great example in biology of hydrogen bonding too, because the dipole moments will allow this kind of weak hydrogen bonding with other, say for example, water molecules. Now I have a picture of it there. So basically here's another water molecule. With their positive and negative dipole moments, and well, maybe that's the hydrogen bond. Okay, this weak attraction between partial positive, partial negative charges. So the polarity of molecules, again, I gave you the general rule. What I had said was polar molecules, like water, or if it's another molecule that's like water in that it's polar, they're hydrophilic. They can interact with it because all the partial charges, it can kind of be soluble in it, right? It's like if you pour olive oil into a glass of water, they don't mix because of this feature. One's polar, water, one's non-polar, fat. Okay. So if you're soluble in it, it means you could dissolve, like sodium chloride. First you can see it, the crystals, when you pour it in. When you mix it up, you can't see it anymore. Okay, it's soluble in it. So basically these molecules made 10 molecules like this, which give them more of a partial positive, partial negative charge. Molecules like this in biology are polar, non-polar, fats, complex ring structures like steroids. Okay, that's what's going to come up for you in A&P are non-polar. That's the general rule. So here are our 20 amino acids. I say which group is the most hydrophobic, and just to remind you, I said probably orange. If you guys kind of have to choose a group. Like for example, here's a phospholipid. I think I defined this the last time. And yeah, so the tail is more, I'm asking the question, which part of the phospholipid is polar? Probably the purple part. Now if you're sitting in the back, you can't see, but it's got molecules like the phosphorus and the nitrogen group that make it more polar. The tail end, which is all hydrocarbons, probably more non-polar. Okay. So the advantage of having a polar head, non-polar tail, let's say, a lot of times they illustrate phospholipids like this. I'm drawing a circle. I mean, if you're a molecule, if you're a phospholipid, you're transported in the blood. Blood is basically water. Half of blood is water, the plasma, the liquid part of blood. If you want to be transported in the blood, you want the head group to be facing out, because that can interact with water. You want the tails to face in, because those are hydrophobic. Now, what I've drawn here, maybe you have cholesterol in here and other fats. Cholesterol, maybe there's some triglyceride esters. We call this the HDL, the LDL, right? This is how it's transported. They're called lipoproteins. I just wanted to give you an example of, well, why do you have one end that's polar, one end that's non-polar? Well, if you want to be transported in the blood, it's good to have a part of you that's polar. You can face the water. Our bodies need a way to transport lipids. Lipids aren't soluble in water. So we use the LDL, the HDL to do. You've heard the LDL is the bad one, right? It tends to clog your arteries. But anyways, you need it. You need to transport this nutrition in your body. All right, so what I like to do now to finish off chapter 2 is to give you specific examples of some of the chemistry. I like to do that because chapter 2, it shouldn't even be there because we have a chem prereq. But it is there, and I like to at least mention the things that come up in 430 and 431 for students. So you say, oh yeah, that's kind of relevant that I needed to know that. So as you go through chapter 2, if you're like, oh, he didn't talk about that, you don't have to know it. Now, if you go through your study guide, are you aware that I have study guides? Raise your hand if you kind of see them. Most of you have. And if you see, oh wait, that's on the study guide, he didn't talk about it, you better ask me. Maybe I forgot, you're still responsible for it. If you see, I'm not looking at notes, right? It's all from memory, so sometimes I forget. But usually not. Okay, so here's an example, a water pore of E. coli. I'm not going to write this one on the board, but you can see these are like anti-parallel beta-pleated sheets. It's a trimer, so that's the coronary structure. The one for humans you should know is aquaporin. That's going to come up in urinary. I don't know if you see that yellow dot right in the middle, in the top figure. That's the pore. It's called aquaporin, so you know it's a pore. No aquaporin. There are different ones. This one is aquaporin 1. It's a water pore. Each one, it's a tetramer, meaning 4 parts, 4 polypeptide chains, identical molecules got a pore in it. Each monomer contains 269 amino acids as its secondary organization, alpha helices, right? So all that terminology should make sense. This is a water pore. Allows the diffusion of water, which is called osmosis. Well, here's a picture of a physiology figure I use in 431. You don't really need to know it now for 430, but just to give you an example. I mean, they illustrate it right here. It's right there. Okay, it's being inserted into the cell. You may not understand what's going on here. This is in the kidney. I'll just give a very brief explanation of what you're looking at. The kidney filters all your blood. You're trying to filter out all the nitrogenous waste that's in your blood. And it's up to the kidney cells to recover, reabsorb all of the water so you don't dehydrate yourself. Because the kidneys filter it like 70 times a day. So all the fluid, all the yellow stuff on this side, we call the filtrate. Because it was filtered by the kidney. And it's on the way out. Once it gets out, it goes down to your bladder and you can't get it back. All right? And most of it's water and other things that you want. I'm just focusing on water. Filtrate is water. It's basically your plasma put it through a filter. It's your blood. The filtrate is your blood. Now, if you don't get it back into a blood vessel, say right here, there's capillaries all over the place in the kidney, you're going to lose it to the urine. So one of the ways the body conserves fluids, there's a hormone you'll learn about called vasopressin hormone, right? An example of a protein. It binds on the cell membrane. There's a second messenger. And it'll insert this aquaporin on this side to like let the water through the cell. And it'll follow its osmosis gradient to the blood vessel. Okay? So that way you don't dehydrate. So you basically just take the water here, insert the little water pore there, aquaporin, and water diffuses through. So you don't dehydrate. We call this reabsorbed water. You don't lose it to the urine. Okay. All right, so that's an example of how a molecule you learned about today could be used in the human body. We also learned about acids and bases in chemistry, and there's weak acids, there's strong acids. Basically, a weak acid doesn't completely dissociate, but strong acids do. And in physiology, acid-base chemistry comes up quite a bit. I'll give you 2 examples. One example is the same kidney. Kidney cell, the exact same thing. Another function of the kidney. Doesn't only just produce urine, does other things for the body. Okay. Usually, a kidney textbook definition will produce about 1 ml of urine per minute. Now the other things it's doing while it's making urine is it helps acid-base balance. The blood should be a pH of 7.35 to 7.45. Okay, the pH. That's why you need chemistry, you know about pH of blood. Now the reason why there's a range is there's different kinds of blood. I mean, you got blood in your arteries, and you have blood in your veins, and those vary a little bit. Venous blood is a little more acidic than arterial blood, but that's the normal range. And to try to maintain that, if you're getting too acidic, maybe less than 735, you want to excrete some of those protons. And the kidney can do that, it's shown there. Here's the amino acid glutamine. Basically, you bust it into parts, and you shove that hydrogen ion onto a molecule like ammonia. It's a buffer. And it takes that hydrogen ion and it excretes it out here, and you get it out in the urine. So basically, I'll just draw it like this. Think of ammonia, NH3, shove a hydrogen on there, acidify it. Okay, and that's the thing you can excrete out of the body in the urine. NH4 plus, that's ammonium ion, right? What you do, coupled with that, is you also reabsorb a base. Sodium bicarbonate, you know, like what's in Tums and other antacids. So by excreting the proton and putting more base in the blood, what you're doing is maybe this helps to counteract acidosis in the blood. Okay, one more example of chemistry and physiology. I'll stop with this. Here's a red blood cell, and it's making a gas exchange at the lung. Okay. Your lungs are filled with air. The tiniest airspace in the lung is called the alveolus. And you have to make exchanges with the pulmonary blood flow into your lungs to get oxygen and to drop off CO2, and that's what's being shown. So they got you a red blood cell there. Here's an RBC. And you can look at the details there, but basically what students are supposed to know is that oxygen, by diffusion, actually I drew that the wrong way. This should be a CO2, not oxygen. CO2 will diffuse into the alveolar space so you can exhale it, get it out of your body. Think of it as a waste product. And it's the O2 that the RBCs pick up. Okay, that's the exchange. Now, if you look at the chemistry, I'm going to zoom in. Do you see the enzyme I told you to note earlier? I had to write it down. If you're not taking notes, then you don't have it, obviously. This one. This one allows this chemical reaction to go fast. So this is the other way, so that you can get, you transport the CO2, not a CO2. Most of it is actually transported in bicarb, but there's a CO2 in it. You just got to chemically convert it back to CO2 when you get to the lungs so you can breathe it off. This is the molecule that can diffuse. All right, well anyways, another example of chemistry and physiology. What I'm going to do now is I'm done with chapter 2. Any questions on today's lecture, or because I'm going to start the next lecture, chapter 3 cell, and then we'll take a break. So I want to talk about mitosis mainly because part of the lab, we're going to look at some cells that are undergoing mitosis. So I want you to be familiar with that. But I have some more general remarks about cells since we're just starting a new chapter. Like I just drew that square, that's a cell. I mean, we kind of take for granted that students know what cells are. Just make sure we're all on the same page. I'll really just kind of get it down to its most essence. What's bigger, a cell or a molecule? Cell. Okay. So just try to. It's hard to get students all these terms. Yeah, you have to think correctly. You have to know what all the terms mean. I call cells the building blocks of life. Your book is just, no, it's chapter 3, cell. They are the smallest irreducible unit of life. There's a point at your life, at some point, you were literally one cell that divided and divided and divided into what you are now. It's kind of hard to believe that we were all one cell. But it was impossible that you weren't one cell. Okay, then you're just a robot, an android. You're not real. But cells, what they do to define a cell, they can grow, they can divide, they have progeny, right? That's why we're studying mitosis. Cells can divide. Now, not all cells in the human body can divide. Most can. Some cells that are really good at mitosis, like for example, stem cells, divide really well. Stem cells aren't mature yet. They got a lot of dividing to do. Maybe epithelial cells, they're known for dividing quite well. The linings of your body. Just to give you a couple of examples. There are cells that don't divide. They're amitotic. Something like some neurons don't divide if you damage the cell body. Nerve cells. Nerve cell regeneration is kind of tricky. I mean, you can look at a figure in your book that says they can regenerate, but then why is there paralysis? Usually when you lose nervous cell function, you've lost that function forever. They don't proliferate, they don't divide again. So they're very important. Muscle, neither. Muscle fibers. Muscle fibers are genetically set. So you can work out and make your fibers bigger, but whatever you got is what you got. Okay? They don't divide anymore. Now, what we study them here in chapter 3 is mostly, think of them as factories. Because when we study transcription, translation, that's kind of the factory function of these. I mean, they can make thousands of different molecules. Okay? Maybe they can produce maybe something like on the order of 500 plus small molecules, maybe 2,000 plus large molecules. Call them macro molecules. Now, I think I have a little list of what I think makes cells unique, why they deserve their own chapter. is how they're able to break down food. Fault number 1. So when you eat and you kind of read the nutrition label, I mean, pretty much what your cells can use for fuel are just 3 things. The carb, CHO, the protein, the RO, or the fat. And that's it. Those 3 things. You know, when you work out, you only use 2 of the 3. Carb and fat. You don't even burn protein during the workout. And what they can do is they can break them down and reassemble the parts into parts that are reusable. Reassemble into usable parts. I mean, I defined all those proteins. Those are parts. Things like that. But it's amazing. You just kind of like eat and all those parts come out. The cell does it all by itself. That's pretty amazing. And also, they can harness the energy released by the breakdown of food and energy. Food is just energy. Okay? It's just, look at the hamburger I sent on the table. It's just energy. It's just in the molecular bonds. It's up to your digestive system to break it down, your body to absorb it and get all that energy out of there. I mean, your body's either going to store it or burn it. That's it. So 2, I will put harness the energy. Released by the breakdown of food. And it harnesses it in the form of, like I said, ATP. There are other high energy phosphates that are talked about in textbooks, something like, I'll put CP, creatine phosphate. That's the C. Okay, so you got that energy. Basically, the cell metabolism that produces these energy-rich molecules is glycolysis. The breakdown of carb. This is a chemical reaction. It occurs in the cytoplasm of blood. And the other one is the tricarboxylic acid cycle. So it should be reviewed. It's also called citric acid cycle. Maybe they're even calling it something else these days. But both of these occur in the cell. The second one occurs in mitochondria. Now, in the mitochondria, right after citric acid cycle, tricarboxylic acid cycle, you have the electron transport chain. I'm not going to go over these chemical reactions. They're most of what the cell does. It's better taught in another class. I'll say that these last 2, you need oxygen as the last electron receptor, so oxygen is required. These are aerobic for that reason. This one's anaerobic. So if you ever heard those terms, they're referring to the metabolism. People also use the word, people love the word metabolism, not necessarily me, but I see it all the time. Over the years, I just come to mean, it just means these, all the chemical reactions in the cell. And this is most of them. Okay, so cells can kind of harness the energy released by the breakdown of food in those reactions. Let's see here. The last one I'll give you is cell signaling. And we're going to see that cells can respond to stimuli. They're not dead. They can respond. This is the entire discipline of molecular biology. We just have to memorize cell pathway after cell pathway. Biochemists have worked out all the cell pathways for whatever. Okay, that's not necessarily this class. You should be aware cells respond to signals. We do talk about it a little bit in endocrine. Okay. So to kind of get back to the notes on my slide, cells respond to stimuli. They have all this metabolism. I just want to focus on the general life of a cell that includes these things, and in particular, the cell division part. Now, if you look at this next figure, it has a nice little graph showing you what's called interphase and mitosis as the life of a cell. Now, some cells that are amitotic will never reach the cell division phase. But most cells that do will go through these kind of general interphase, mitotic phase, and when I get to the mitotic phase, I'll really focus on the different phases of mitosis, since that's something we can observe in the lab. Interphase is most of the cell's life. Kind of what I call the heyday of the cell. The cell is kind of like doing what it's supposed to do in its tissue. You know, if it's an epithelial cell or a gland cell, that's G1, the growth phase. So I'll put it under there. Cells made, function accomplished. If it's a cell that's gonna divide, then that's it. It starts to make preparations to divide. For example, S phase. It says growth and DNA synthesis. What I'll just say is S phase, I put DNA replication. If one cell is going to become two, you got to double it. So two daughter cells will have the exact same amount of material, just the right amount. So that's something the cell would do if it's like gonna get ready to divide. Okay. The other thing, G2, making your final preparations here. Let me read what it says on the slide. It says growth and final preparations for division. Very, very generic. Basically, you have to double everything. Let's look at this top cell here. cell, it's nuke, it's purple there. They draw what are called the centrosome. Let me go ahead and define that now. That's one of them. There's two in the picture there. Let me define what this is. I'll say centrosomes replicate. What a centrosome is this thing. Let me draw it out here. A centrosome is two centrioles. Those two are usually perpendicular. And there's a bunch of like rays coming out of it. Now those astral rays, as they're called, they're basically just other molecules. They're called microtubules. And in fact, microtubules make up the astral rays and the centrioles. The whole thing is microtubule. But individual microtubules are the astral rays. Okay, and that whole thing is a centrosome, right? The whole thing. The centrioles with the astral rays coming out of it is a centrosome. So part of this class is just knowing what the words mean. In fact, I would say that's most of it. If you don't even know what the words mean, I can't work with you. So I have to do my job and just at least tell you what the words mean. Now the reason why I mentioned that is don't get frustrated. If you're frustrated, it's probably because you keep reading words you don't know. So you have to like look it up. That can take hours to read like 2 paragraphs. You keep looking up the words you don't know. That's the process. Okay, trust me, I've been doing this my whole life. You got to know what the words mean first before you try to increase your understanding. Centrosome, centriole. Okay, that's it. Microtubule, astral ray. This is very important for the cell division. The astral rays will actually attach to chromosomes and move them around. Now, when a cell gets ready to divide, they replicate. That's how you know you're kind of getting ready to enter cell division. So I draw 2 of them. So you see 2 of them, you know you're like kind of late in the G2 late interphase, getting ready to enter cell division. And let's take a break, and we'll actually talk about that when we come back. Remember, take everything you need, I'll lock the door, come back in 15 minutes.